will have a function of that gene will encode for the mRNA. mRNA will become a template to make amino acid, to make protein. So if this area of the difference here will actually get passed on to the other cells, and that would actually happen at the region which is the affecting on the expression of mRNA, making the template of the mRNA change in a sense that is disrupt the codon. So you may actually have the abnormal gene expression. Okay, I will talk a little bit about that in the lecture protein. Then we will touch on the translation as well. So these are the DNA repair mechanisms. I won't go into detail, but talking a little bit about each one. So we have the touch on the post-replication mismatch repair already, where you have the replication going, and then they have the mismatch, and you have a set protein to detect the methylation pattern in eukaryotes. In eukaryotes, they detect the NIC, which I didn't mention on that, but anyway, that one will detect that, okay, we have replication going on, we have error in replication going on, and then they create the endonuclease activity to cleave it and take it out, and then making new nucleotide to fix that one. The other two here is the double-strand break DNA repair. For some instance, you have a very strong ionizing radiation exposed to the ionizing radiation. It may create double-strand break. Or some instance, you have the reactive oxygen species, and that will make the mutation happen. And some mutation is actually very bad in the sense that it stores the replication fork. If you remember that the fork is going like this, right? And then sometimes, if you have mutation, the DNA polymerase can't go, and then sometimes it stops. The stall is sometimes in worst case is introduced double strand break. So when you have double strand break, you start having some protein in the sense that, oh, if double strand break happens and we need to fix it. So these two are the mechanism that will fix the double strand break in our cell. It depends whether you have a homologous region in the genome, or at which state of the cell cycle you are, and then they will pick that which mechanism that will use to take care of that double strand break. So for the nucleotide excision repair, so that is the mechanism that will help with the abnormal structure of the DNA. Example that I gave you was the thymine dimer from UV exposed, and that make abnormal structure. It's triggered the nucleotide excision repair to have endonuclease activity to cleave the full upstream and downstream of that position, having helicase to remove, have polymerase to add a new base, have ligase to finish that off. Okay. So that is sort of the diagram that will give you an example that at which damage that will trigger that which pathway. See, I don't want you to remember details, but I like to appreciate on the different kinds of the DNA damage will actually trigger the different mechanism. The key of the DNA repair will be that you have a sensor that will sensing different type of DNA damage on the DNA. Some can be single strand break, some can be double strand break, some can be the base damage like apurine from the depyrination, and some could be a very bad mutation or cluster lesion. From that, you will have a protein to stop cell cycle. You have cell cycle arrest to give enough time for protein to repair by those mechanisms. And if the repair going to success, they will have a trigger saying, okay, you can go on with the cell cycle, and then it can move on. Okay. If not, we would actually have some protein that will trigger the process called apoptosis. The protein that will involve in that a lot will be p53 protein that you may have heard of, because the mutation on the p53 will actually lead to about 50% of all cancer that will actually found in human. Okay. Type of base change and mutation. So it could be the mutation that could happen. It could happen just the base change from one base to the other, from AT to GC, or it could be CG to GC. So it just called a different name, but transition or transversion, whether it's actually going from one type of base to the other. One of the important one that sometimes is actually making effect to cause human disease, it will be for the missense mutation, that you will have a mutation that change a set of codon to encode one amino acid to change to the other. So that will be one of the key ones. Some other one will actually have base change that make base to create a stop codon. For example, we have AAA here, which will encode for lysine. But when you have a mutation that change that A to be T, effectively you change that codon to be UAA, which that will become a stop codon. That type of mutation is called nonsense mutation, because sense is making mRNA. When you have something change and making no mRNA, so it's making nonsense. So that's nonsense mutation to make it a stop codon over there. And some mutation will actually cause the frame shifting and the G1. So just the base here have the change of the deletion of the one base, and then you shifting a frame and then making new set of amino acid. Normally for the frame shifting, you change amino acid for certain number, about 50 peptide amino acid or polypeptide, and then you run into stop codon eventually. Okay, I think I will leave this one for the protein lecture. I'm just going to skip that. Okay. Now we sort of talk about type of mutation, right? It can be the type that have base change, or some small insertion, some deletion, and some base change will lead to missense mutation. Some will lead to nonsense mutation, right? If you have missense mutation and nonsense mutation, so I just want to maybe just throw you a little question that how should we detect them? How should we detect the mutation? First. Second, if we have small insertion deletion, how should we detect them? Any idea? First base change, just from A to T, A to G, like that. The other one is like just one base missing, or 5 base missing, or 10 base missing, or the whole 1000 KB missing, 1000 base pair missing. So how should we detect them? Any idea? Anyone? being exposed to molecular technique before? Want to give me some answer, clue, please? Any answer? PCR. Okay, what's a PCR? PCR. What is a PCR? Just a little bit extra. Polymerase chain reaction. So we can actually amplify the DNA up, right? So we can actually take material of interest. For example, we can have white blood cell, we can have DNA, extract DNA, amplify the DNA up in the specific region, right? And then how we detect the base change. Is that for base change or is that for the small insertion deletion? And we look at the difference by size, because now we're talking about, okay, for example, if we have interest into this region, we design primer to cover the region, right? Amplify it up from 1 copies to millions or 100,000 copies. And then if you look at the amplicon is the amplified product of this specific region. You can tell the size difference of the amplified product, right? So PCR and then what? Because you can't just like PCR in the tube, and then you need some way to tell, right? Gerontophoresis. Okay, mention gerontophoresis. Gerontophoresis is the technique that you put your DNA that you need to amplify it up first, right? Because just few copies you may not be able to visualize it, right? And then you need to amplify it up using PCR, and then we run it on a gel just to make them separate by size. So if you design the primer in this particular region, and then you have size differences from the different deletions, so you will see fragment with different size, right? So that would be one thing that can be detected. But would that work for base change? If we amplify it up, just a PCR, and then you have base substitution, would you be able to tell the difference? No. What could be the things that we can do?
PCR IFLP.
PCR IFLP. What is a PCR IFLP?
After do the PCR,
yes.
When we have the DNA product, we use precision enzyme to cut the DNA at the specific size. If maybe if the white height we detect can be cut by the enzyme, and when there are the mutation at the site, reticent enzyme can cut. The reticent enzyme cannot be cut the mutation one.
Okay, so your friend just mentioned that other than this small insertion deletion, we can look at the base change as well by designing the sequence of interest. For example, if you amplify the region up, and you have base change. Maybe I'll flick onto that slide. Okay. So if you have a product of interest which is about 210 base pair here, that could be a wild type product with a base at particular position that we look at, like this one. And for the mutation, you have a single base change from G to C here, and that if that create a cut side of the enzyme, of the restriction enzyme, when you amplify them up of the control Y type, and maybe the positive control for mutant will be even better. And then you have a sample of interest and then put them together. You can see that whether the Y type, you will have only the uncut product here at 210. When you have a mixture of Y type and mutant, you will see two bands, right? When you see two bands like this, if the mutation is a germline mutation, the germline mutation means that in every cell of the body entitled to mutation, right? Which means that you take any DNA from the cell, 50% will be Y type, will be normal, and another 50% will be mutant. You will see something like this. But if you see just only that cleaved product like this one, so that will be fully mutant. But if you have extra band here, what does that mean? Heterozygous, right? You have one allele having the uncleaved product, and the cleaved product is like that one. So that is the PCR-FLP, which based on PCR and restriction enzyme. So what is restriction enzyme? Restriction enzyme is the enzyme that will cut piece of DNA through a set of specific sequence. It's called palindromic sequence. When you look at the palindromic sequence like this, when you read from one side to the other, and when you go on the other side as well, it will read the same. It's GAATTC and GAATTC. Okay? It might be hard if you have to look through all these yourself. But actually have a program that you can actually throw the sequence into it and then give a prediction for the restriction side. All you have to do is you throw in the Y type piece of amplicon and also the mutant piece of amplicon and identify the difference of the cleavage side by the program. And then you can set up the assay and then you can detect this small change. Okay, so we have PCR, looking at the size by amplifying a specific product of particular region in the genome, in the gene, and run it on a gel to see the size difference for the small mutation. You can also look at the base change by designing PCR IFRP. Are there any other molecular technique that you guys have exposed to? Sorry? Sequencing. What is sequencing? Have anyone heard of sequencing before? Okay. So sequencing, we can talk about classic sequencing of the Sanger sequencing, where you have a mixture of the incoming or the adding nucleotide, because all the enzyme does is adding new nucleotide into the newly synthesized strand, right? As I mentioned, to make polymerase function properly, you need a template for show of nucleotide and 3' OH, right? In the reaction, what it does is it provides a mixture of the dideoxy and the deoxy ATP into each lane. So if the enzyme randomly pick the template, pick the nucleotide with the 3' OH, they can continue with the polymerase reaction to continue the longer strand, right? However, if the enzyme randomly pick the dideoxy nucleotide into the last base, it can't continue the polymerase reaction. And if you separate each lane. If you separate each lane of the A, T, G, C with a mixture of dideoxy A and normal A, dideoxy T and normal T, dideoxy G and deoxy G, you will have a random continuation of the G, and then you randomly pick the G that have the dideoxy. It will stop here, there, wherever it does have G. C, sorry, because there is template, right? So that should be on top, okay? So that is the G there, and G there. So if you run that on the capillary, or you run that on the gel electrophoresis, you will see the size different, right? That is a classic one. However, if you have dideoxy that is actually tagged with different fluorophore, fluorophores mean that the color that can be activated by fluorescence will give you fluorescent signal. So you can have detector at different wavelength to detect different nucleotide. All you have will be the product that will actually having a different color once it actually run through to the capillary here. And the end product of what you're having here would be the sequence of what you sequence on. So rather than you get like the RFLP to